try and, well, we'll definitely finish up with the effects of different environmental factors on bacterial growth. And so I left with the oxygen relationships of different types of bacteria. And one thing that's kind of, well, useful to know, I think, and interesting is how do you culture strict anaerobes in the laboratory? Because most of the time, when you're working with bacteria, one of the things that you want to do is spread them on an agar plate and get isolated colonies. Starting with an isolated colony is an essential step in bacteriology because it will give you a pure bacterial culture. Every cell in the colony derives from one initial clone, right? So if you pick a colony, all the cells in from that colony are, clones of each other. And this is what gives you a pure culture, pure bacterial culture, also called an axenic culture, right? So this is an important step in isolating bacteria and studying them. But how can you do this with an anaerobe? Because, of course, on the surface of the agar plate, it's in contact with the air. And if you're working with a strict anaerobe, the oxygen in the air will be toxic for them and kill them. So what is often used, or is the most convenient and the cheapest way to do this, is to use an anaerobic chamber. And you can see the setup here. You've got some kind of jar, and it's closed tightly, hermetically, with a lid with a rubber seal at the top. And you can stack up the agar plates that you've inoculated, and there's a kind of pack of chemicals. And you add some water into the pack. The chemicals will start bubbling up when you quickly close the lid of the jar. And what's going to happen is that the chemicals in here, so you have sodium borohydride, a mixture of citric acid and sodium bicarbonate. And when you add water to them, there are two chemical reactions that occur that release hydrogen gas. And there's also a catalyst up in the lid of the jar, which will catalyze the reaction between hydrogen and oxygen to produce water. And that will use up all the oxygen gas that is present in the atmosphere here. So you create an atmosphere without any oxygen. And then even strict anaerobes can grow in this kind of setup. Okay, so that's enough about oxygen, I guess. The next environmental condition is pH. I didn't change this. So most eubacteria are neutrophile. That is, they prefer to grow at neutral pH plus or minus 1.5 pH units. Next, well, and apart from that, there are, of course, bacteria that prefer to grow at acid pH, which are non-surprisingly called acidophiles. And bacteria that prefer to grow at alkaline pH are alkalophiles. Now, among acidophiles, I didn't really put an optimal pH down here because it's quite variable. There are plenty of bacteria like lactobacillus. Their optimal pH for growth will be something like 4 or 5. Maybe, 4, 5, 6, something like that. 4 or 5, let's say. But there are, extreme acidophiles which will grow better at pH 2, for example. So once again, when these are in blue, these are archaeobacteria, which can be found in extreme environments, very, very acidic lakes or hydrothermal, sources, hydrothermal vents. Lactobacillus, rather more common. So these will be bacteria that live by fermentation, produce acidic byproducts, and they like to live in this acidic environment. But their pH optimum will be something a little bit more reasonable. Alkaline loving alkalophilic bacteria, once again, you can find them in unusual environments, lakes which have an alkaline pH. Now, for neutrophils, then whatever pH they're growing at, the cytoplasm will remain at neutral pH. So they need to have some kind of homeostasis in order to maintain their cytoplasm at around pH 7, 7.5, even though outside it may be more acidic or more alkaline. And that's certainly true of these, fermenters, which will produce acidic byproducts. So they'll have a tendency to acidify the external medium, but in order to survive, they have to keep their cytoplasm at neutral pH. So for Escherichia coli, this is the classical example where we know pretty much everything about how this bacterium lives. If it's growing in acidic conditions, what the problem is for the bacterium is that there'll be a tendency for the pH to equilibrate across the membrane, and protons will have a tendency to accumulate in the cytoplasm and acidify the cytoplasm of the bacterial cell. And that's going to be something which is very bad. Why? Because if you think of all the proteins in a cell, they need to stay soluble in water, right? And in order for that to happen, they need to have some groups which carry a charge on the outside of the protein surface. So the carboxylate groups and the amine groups that are exposed on the surface of the protein, they need to be in the correct charge state so that the whole protein will remain soluble in water. And if the pH changes, then these proteins can suddenly become a lot less soluble and precipitate. And that will be very, very bad for the bacterium, right? So if protons are having a tendency to, diffuse into the cytoplasm of the bacterium, the E. coli here needs to get rid of them, to export them. And one of the things that can be done is for the proton-dependent ATP synthase to work in reverse. So normally this ATP synthase is used to generate ATP by importing protons. But it can also work the other way around, hydrolyze ATP and export protons. So the energy is used here in the form of ATP to keep the pH more neutral and less acidic. So that's one technique or one mechanism used by bacteria to control their internal pH, and they will also express chaperone proteins to try and avoid protein denaturation and precipitation. Well, what happens if the opposite is true and these bacteria are growing in alkaline conditions? Well, in this case, the tendency will be for the cytosol to become too alkaline. So they need to try and retain protons and import them to acidify the cytoplasm to try and keep it balanced. So in this case, what they can do is import protons and export sodium. And once again, this will cost energy because the protons that are being exported here, they'll have to be re-imported somehow with another kind of transport system, and that will cost energy. So import protons when the conditions are alkaline, and once again, chaperone proteins to prevent protein denaturation and precipitation. And the other thing that bacteria can do to kind of compensate for external alkaline conditions is to change their metabolism a little bit, produce more fermentation products which will tend to acidify the external medium. Okay, so that's pH regulation. Next thing is sodium chloride, salt concentration. Once again, most bacteria prefer salt concentrations to be not too high. Okay, so for bacterial growth, they always need some NaCl in the external medium. You can't grow bacteria in pure water. But most bacteria will have a growth optimum of around about 0.2 molar sodium chloride. And that's not too far off an isotonic salt concentration, right? So inside our cells, sodium chloride concentration is what? Anybody know this one? Isotonic sodium chloride, sometimes called in French sérum phi. Nobody has any idea of the number? Probably, I guess, about 142 millimolar sodium chloride in our cells. Something like 150 millimolar, okay? So bacteria will like to grow, their preferred growth concentration of sodium chloride will be about 0.2 molar. A little bit, just a bit higher than isotonic. So that will be their preferred concentration of sodium chloride, and most bacteria can carry on growing up to about 1 molar salt concentration in the medium outside, which is not the case for us. I mean, like, we would not be happy if you're swimming around in a bath of 1 molar sodium chloride. So, just by comparison, seawater is about 0.6 molar, which is about 35 grams per liter. Above 1 molar sodium chloride, most bacteria won't grow, okay? Apart from halotolerant bacteria, halophiles, and extreme halophiles. So halotolerant bacteria, their optimum sodium chloride concentration is 0.2 molar, just like non-halophilic bacteria. But they can grow up to about concentrations of about 3 molar. So the big classic example of this is Staphylococcus. So if you remember from the culture media, Chapman medium is selective for halotolerant bacteria, including Staphylococcus, because it contains 75 grams per liter of sodium chloride, which is probably about 1.5 molar. So it's too high for most bacteria to grow. Halotolerant bacteria will grow on culture medium with 1.5 molar sodium chloride. Now, there are also halophilic bacteria. So the difference between halotolerant and halophilic. Halophilic bacteria don't grow very well at concentrations less than 0.5 molar sodium chloride. So, and they prefer concentrations higher than 1 molar. So an example of this is Ectothiorhodospiro, which is a purple sulfur bacterium. It's a photolithoautotroph, which grows in environments with a lot of salt. And extreme halophiles like Halobacterium, once again, it's an archaeobacterium. It's an extremophile. They can only grow in lakes with very, very high sodium chloride content. Okay, so once again, most non-halophilic bacteria, they can grow in salt concentrations over quite a wide range, okay? So they can grow from about, 20 millimolar, 50 millimolar salt up to about 0.5 molar. So, a tenfold range of salt concentrations. But during that time, they have to keep their cytoplasm at a pretty much constant sodium chloride concentration, and they need to retain water inside the cytoplasm. So they have to have some means to, not absorb too much water and not get too dried out. So if bacteria are growing in hypertonic conditions with a high concentration of sodium chloride outside of the cell. The risk for the bacterium is that all the water inside the cell is going to diffuse out. And then the whole thing will be too dry and it will stop working, stop running. So the way that many bacteria respond to this is by accumulating or increasing the intracellular concentration of what are called compatible solutes. And these are small organic molecules that have a charge. So they have some kind of ionic strength as the concentration increases. So amino acids like proline, glutamate, and then some things like glycine and betaine, which is this thing here. Okay, so glycine is, if this was NH3 plus, that would be glycine, right? An amino acid, except it's got these methyl groups on the nitrogen here, so it doesn't really change the pH very much, the concentration of this molecule. So all of these small organic molecules that carry some kind of charge, they have the ability to retain water. Okay, so that's how bacteria will kind of react to growth in higher sodium chloride concentrations. Now, extreme halophiles, that's not enough. They have some kind of extra tricks, like a halobacterium will accumulate a high concentration of intracellular potassium to kind of compensate for the high concentration of sodium outside the cell. Now, in hypertonic conditions, the problem for the bacterium is the opposite. So in hypertonic conditions, water outside the bacterium will tend to diffuse across the membrane into the bacterial cell, and that will cause osmotic pressure inside the bacterium. So as we all know, right, the peptidoglycan layer, the cell wall, is pretty strong. Its function is to, keep the cell intact despite osmotic pressure. But there's a limit to every kind of biomaterial, and if osmotic pressure is too high, this can really cause problems for bacteria, especially if they're growing, okay? Growing bacteria, they express autolysins, their peptidoglycan is being broken down and resynthesized. So if they've got too high osmotic pressure, this can cause them to explode. And you wouldn't want that to happen, right? No. So what they do is to try and reduce the osmotic force inside the cytoplasm that's pulling this water in. So instead of having these compatible solutes as simple monomers, they'll polymerize them. So they'll be polymerized into inclusion granules. And these don't exert osmotic force. And the other thing that they can do is if the osmotic pressure is too high, there are mechanosensitive pores in the plasma membrane which will open if the stress is too high and let solutes come out. Salts, amino acids, this kind of thing, so that they can survive very, very low salt concentrations outside the bacterium. Okay, so the salt concentration outside the bacterium is kind of related to another important environmental factor in bacteriology, particularly in food microbiology, which is what's called the activity of water. Now, I try to understand this in an intuitive way. I mean, it can be expressed in physical-chemical terms, but the way I understand it is that, okay, if you've got a particular volume of pure water, okay, you've got a certain number of water molecules here, and all of these water molecules are free. They're able to interact with other things, and they're able to, evaporate at the surface of my rather rectangular-shaped water droplet here. However, if you've got some kind of macromolecules in the same volume, and I took special care to have exactly the same number of water molecules here, so the amount of water is the same in the same volume, But you've got some macromolecules here, and they are interacting with these water molecules because to keep macromolecules hydrated, some of the water molecules will be interacting through hydrogen bonds with charged residues, hydroxyl residues on the macromolecule. So these molecules of water here won't be available to interact with some other kind of chemical or biological system. And they won't be able to readily diffuse or evaporate from the surface of my water droplet here. So even though you've got the same amount of water here in these two droplets, in this one on the right, it's not all available, okay? And the less available water you have, the less it can evaporate off from the surface of this droplet. And that's what allows us to measure something called the activity of water in any kind of substrate. Can be like a solution with high concentration of salt or glucose, or it could be a plate of porridge, could be a yogurt, could be a biscuit. Okay? All of these things, they can have an activity of water, which can be measured because it's basically 1/100th of the relative humidity that is released by this substrate. Okay, so that's the definition of the activity of water. So before we go on to understand why this is important for microbiology, I just want you to think about this. I don't know if the link will be working, so just think about it and we can do it by a show of hands maybe. Okay? Okay, let me just check. I can probably put the right QR code in there in a minute. Should have done that before, I guess. Oh, come back. Ah, come on. Whoa. Okay, so this will work. Taking too long. Okay, everybody finished with this? What do you want to do? You want me to show this, or are we going to go by show of hands? Because not many people sent in answers, to be honest. Show of hands, everybody okay for that? Yeah, hands up when it's the right answer. Okay, so true or false? The scale goes from 0 to 1. Who says true? Half the people. Who says false? It's true. Because, slide before, the activity of water is 1/100th of the relative humidity. So the maximum for relative humidity is 100%, right? That's the amount of water vapor you'll get released by pure water. 100% humidity, you can't get any more. It's at equilibrium. So the scale, and dry, no water vapor is 0, right? So the scale will go from 0 to 1. Is equivalent to water content. Who says yes? Who says no? Yeah, you're right. It's not the same. It's kind of related, but it's not the same thing. Like in my example just before, I had the same number of water molecules in the volume, but the activity of water was different. Okay, activity of water is higher in a high salt solution. Who says yes? It's true. One person. Who said no? It's not true. Two or three people. It's the no people. The no people. You're right. It's the other way around. Okay. So the more salt you have in the solution, the less water is going to be available for other things. Right? Because it's too busy packing around the sodium and the chloride ions. So the higher the salt concentration is, the lower the activity of water will be. That's why these two things are related, okay? And so that's important to understand the way round it works, okay? And question 4, who says yes, this is definitely true. This is totally true. It's like well, that's all they do. They're doing this 24/7. They wake up in the morning. What other crazy thing can I do to annoy biologists? But like most of the things that the chemists invent, it might be annoying and complicated and difficult to understand, but very, very useful and important, okay? So why is this important? Because just like the ability to grow in high salt conditions, the ability to grow at a low activity of water is, different for different types of bacteria. So in a kind of fresh food substance, like fresh fruit, meat, blood, vegetables, fish, something like that, the activity of water is very high, okay? 0.98 to 1. So there's always free water around for bacteria to grow. So pretty much all gram-negatives and gram-positive bacteria can grow at an activity of water higher than 0.98. Lower than 0.98, most gram-negative bacteria don't grow. Okay, only gram-positives will grow. And so most gram-positives can grow down to an activity of water to about 0.95. From 0.95 down to 0.9, most gram-positive cocci can still grow. But lower than that, pretty much only Staphylococcus, halo-tolerant Staphylococcus. They can still grow down to an activity of water about 0.85. And that's pretty much it for bacteria. Okay, lower than that, you can still get some fungi which will grow at lower activities of water. So this is very important for food conservation. Okay, so any kind of fresh food, bacteria, pretty much all bacteria can still grow on it. Okay, things that are conserved with drying them out, getting rid of most of the water, where fewer and fewer bacteria can grow on them. Something like a salami, Staphylococcus can still grow, but most bacteria can't grow on sausage like this. And then when you get down to things like, cereals, I don't mean cornflakes, I mean wheat, oats, barley, the grains. Most of the water is gone during, before harvest time. When it's sunny, the grains dry out, right? So the activity of water is very low. That's why they can be conserved. If it stays dry, they won't go off. Okay? So very important for food conservation. And, it tells you pretty much what kind of microorganism is going to cause you a problem for a particular kind of food. So that's why storage of cereals, storage of grains, the big problem is not bacterial contamination, but spoilage from fungi, okay? So they can get mold growing on them. And then once you get down to something like this, dehydrated foods, like instant noodles, something like that, the activity of water is less than 0.6, so no microorganisms can grow on this. And they can stay stable at room temperature really for years. Okay, so these things, they all come together in the way that different food products are conserved. So if you've got something like, something's going terribly wrong here. Okay, so bread like this, you can buy in the supermarket. The activity of water will be about 0.95, maybe a bit lower, actually. But so it's low enough for most bacteria not to grow. The risk is going to be contamination from fungal spores. and molds growing on it. And that's prevented by packaging this kind of product in an atmosphere that doesn't have any oxygen. Okay, so you don't just have air in here, right? You've got a mixture of nitrogen and CO2. Now, fungi, they are obligate aerobes, so they don't grow in the absence of oxygen. So these two things will be enough to conserve the product at room temperature for several weeks. Now, sometimes if you see one in the supermarket, normally you don't see them in the supermarket because the employees come and take them off the shelves. But sometimes you can see some where they've got a little bit of mold growing on them, right? And that happens when the packaging is damaged. So the air gets in from the outside, and suddenly any kind of spores that have contaminated the bread during the baking process, they can grow. So that's why keeping the packaging intact is very important for the shelf life. So this kind of thing is another miracle of food technology. Anybody have any of this at home? I've got some at home. Anybody eat some of this regularly? No. So my dog eats stuff like this. Okay, so this is dried dog food or cat food. So that's got an activity of water about 0.65. It can stay stable at room temperature for several weeks. No microbes can contaminate this. There's not enough water around. So other things that can stay stable for a long time, things like cheese, the activity of water about 0.95. pH is acidic, so neutrophils can't grow in there anymore. And in the inside, there's no oxygen because it's all been used up. And the crust on the outside will prevent contamination by anything else. So this kind of thing will stay pretty stable for several months, in a fridge or in just a cellar, 10 degrees, 15 degrees. And you have other fermented food products like yogurt, okay, pH 4 to 5. Most bacteria won't grow, it's too acidic. Some bacteria can grow, of course, and molds can grow on the top of this. So it needs to be conserved at 4 degrees because the activity of water is higher. Plenty of water in yogurt, loads of things can grow in there. Okay, so that's the importance of these environmental conditions for bacterial growth. Oxygen relationships, temperature, activity of water, and pH. Okay, you put these things together, it determines how long a food product can last and under which conditions. Okay, the last thing before we move on to bacterial metabolism is radiation. Bacteria can be exposed to different types of radiation. Ionizing radiation from the radioactive decay of some rocks. If you live in Brittany, the granite is we're releasing radon. You get a little bit of dose of radioactivity, and then of course any bacteria that are growing on the surface of the planet are exposed to sunlight, and some sunlight contains ultraviolet. Well, sunlight is composed partially of ultraviolet radiation. So these are all more or less toxic for DNA. Ionizing radiation, UVB and UVC are all toxic for DNA. UVA, not so much. Okay, this is the good one. It helps you get a tan. This is the bad one. Will cause you cancer. So what have I got to say about this? Okay, so all bacteria are more or less exposed to these things, especially if they're living on the surface of the planet or if they live, in an aquatic environment near the surface. So they need to have some kind of means to defend against, the damage caused by ultraviolet radiation. So they'll have enzymes for repair of DNA. And it's also kind of related to oxygen tolerance because the effects of ionizing radiation and UVB are indirect. They induce the formation of reactive oxygen species, and these are the things that are going to be chemically reacting with DNA molecules. UVC but we don't have any on the surface of the planet. Outside in space, yes. And this is directly absorbed by DNA, and DNA is directly damaged by UVC. And it's extremely toxic and dangerous for any kind of life. And the only reason we can walk around and live on the surface of the planet right now is because the ozone layer absorbs all of this. So why is this useful for us? So ionizing radiation can be used to sterilize objects that can't be sterilized by heat, by autoclave. So anything made out of disposable lab plastics, injectable products, they can be irradiated, and that way we know they're sterile. UVC radiation can be used to sterilize water. I don't really know in what conditions, but it does exist. Okay, so that's kind of the summary of what we've seen here, but I don't really want to go through these one by one. This is just, the summary sheet for when you're revising, learning this stuff. You have to know all of these things. Okay, so that leaves us about 40 minutes to move on to I'm going to change the order here a little bit. The last two classes, which are going to be about bacterial energy metabolism. And this is related to these two characteristics of bacteria. They can live on anything, right? They can perform unusual biochemical transformations that allow them to live on a weird variety of substrates. And that's kind of interesting and useful for us because we can use what bacteria do in industrial applications. So that's the point of understanding bacterial energy metabolism. Now, what we're going to do is first start with perhaps the simplest way of getting energy, which is to produce or extract energy from organic chemicals without any oxygen. So these bacteria will be organoheterotrophs. They start with an organic source of carbon as their source of energy and electrons, and they will not be using oxygen. So they'll be using fermentation to derive metabolic energy. That's what we'll do first, and then we'll do the more complex stuff, electron transport and respiration, and then how do chemolithotrophs and phototrophs derive energy. Okay, so just to start with this. Now, I guess you probably all know, but it's worth having a reminder that there are three types of energy currency in cells. So ATP, NADH, NADPH, and the proton motive force across a membrane. And bacteria, they need all three of these things. Okay, so ATP, the hydrolysis of ATP to ADP plus an inorganic phosphate, releases energy which is useful for all sorts of biochemical reactions inside cells. And interestingly, it's not just ATP, but the other nucleotide triphosphates are also used to, as cofactors for important biochemical reactions. So protein synthesis on the ribosome doesn't use ATP, it uses GTP. Lipid synthesis, again, bacteria need to put in energy to produce these polymers. This comes from cytosine triphosphate. And polysaccharides, it's uridine triphosphate. If you remember peptidoglycan synthesis, we had UDP-NAM at the beginning of it. So all, polysaccharides, oligosaccharides, they all use UTP. And I don't really know why, okay? I always found that fascinating, but it's true. It's true in bacteria, it's true in all forms of life. Membrane transport, ATP binding cassette transporters require ATP. NADH, why is it necessary for bacterial cells? Well, bacteria need to perform a lot of reduction reactions in their metabolism. So, of course, bacteria that are autotrophs, if they have carbon dioxide as their only source of carbon, they need to transform it into carbohydrate. So this is a reduction reaction, okay? Carbon dioxide is going to be reduced to carbohydrate. And a reduction is a transfer of electrons. And the reducing agent here will be NADH or NADPH at different steps in the biochemical pathway. The same is true for production of fatty acids from carbohydrate. It's reduction, and you need to have a source of electrons to fuel this redox reaction. Also true for the assimilation of nitrate into amino acids. First, the nitrate's got to be reduced to ammonium ions, and then this ammonium can be introduced into amino acids. So that's essential for any type of bacterium. Need a bit of NADH. The other thing is, in bacteria that perform respiration, NADH is used to generate the proton motive force. Yeah, that's true. And proton motive force, what is that useful for? Membrane transport. These proton symport systems require the proton gradient. As we saw, movement from the flagellum, that requires a proton gradient. That's what gives the energy to rotate the flagellum in the membrane. And in a lot of bacteria, the proton gradient is used to generate ATP by the proton-dependent ATP synthase. So, as I said before, as we saw just now, in conditions of acidic conditions outside the bacterium, the proton-dependent ATP synthase can work the other way around. So that also means that this is a kind of reversible process. If you've got ATP, you can make a proton motive force. If you've got proton motive force, you can make ATP. So there's these kind of types of energy currency are kind of interchangeable to some extent. So if you've got one, you can make the other ones, okay? But you need all three of them. Okay, so that's what the objective is going to be for bacteria. So firstly, okay, how to get metabolic energy, NADH and ATP, from organic carbon in the absence of oxygen without any or any other inorganic electron acceptor. So we're going to start thinking about glucose, sugar fermentation. Now, there are other fermentation pathways, but let's just think about fermentation of sugars to begin with. So fermentation of sugars starts with glycolysis, which is the transformation of glucose into pyruvate. And then afterwards, the pyruvate is going to be used for the fermentation pathway. Now, there are three pathways of glycolysis, which are the Embden-Meyerhof, the Duderoff, and the pentose phosphate pathways. So the Embden-Meyerhof pathway, which I guess you haven't seen yet, it's in biochemistry, but you guys haven't done it yet, right? No, well, that's what I think. So just in summary here, okay? One of the things you have to remember is how many carbon molecules do I have? Okay, glucose, how many carbons? 6. Right, start off with the 6 carbon molecule. So the first thing that's going to happen is that glucose will be phosphorylated, glucose 6-phosphate, transformed into fructose 6-phosphate, and then phosphorylated again to fructose 1,6-biphosphate. So this step doesn't generate energy, it costs energy, right? You've made an investment of 2 ATP molecules. But you're going to get the money back later on, okay? Don't worry. Then what happens is that this fructose 1,6-biphosphate is split in 2 to give you 2 molecules of glyceraldehyde 3-phosphate. So 2 3-carbon molecules, each one with 1 phosphate group. Okay, this is 6 carbon, 2 phosphates, 3 carbon, 1 phosphate. The next step is where you get your money back, okay? Because the next enzymatic step will result in the phosphorylation of glyceraldehyde 3-phosphate by an enzyme called glyceraldehyde phosphate dehydrogenase. So you've got two phosphate groups on this very small 3-carbon molecule, and then they're taken off. And each time you take one off, you can generate 1 ATP. So the net yield here, if you count them up, is for each molecule of glucose, you get 2 molecules of NADH, okay, because this is all multiplied by 2. You get 2 of these molecules per molecule of glucose. So you get 2 NADH. Here you generate 4 ATP, but you've spent 2, so your net yield is 2 ATP per glucose. And the pyruvate is the end product of the pathway. Now, one of the things that is important, and in biochemistry they'll beat it into your brains like without mercy and without any kind of compassion. And what they're going to tell you is that this is called substrate level phosphorylation. Well, that's it. That's the technical term for what is happening here, because you get this substrate which is phosphorylated. Now, the enzyme that does this is glyceraldehyde phosphate dehydrogenase, and it's incredibly important because this is where you get something for nothing. It's incredibly rare in life that this will happen to you, right? There's no such thing as a free lunch, except if you have glyceraldehyde phosphate dehydrogenase. Because what is happening here is that there's a favorable redox reaction, which is coupled with the phosphorylation. And that allows you to get one phosphate group on here for free, okay? And then you can take it off and generate ATP. Okay, so that's the Ender-Meyerhof pathway. The other two pathways are something similar. You don't have to try and learn these at all. But the only thing that perhaps is interesting to notice here is that the pentose phosphate pathway will expel one molecule of carbon dioxide, so you end up with a 5-carbon sugar here. And that means at the end you've got acetyl phosphate and pyruvate. The pentose phosphate pathway is, most organisms have it. I mean, we have it, but it's not used to generate energy. It's used to generate, ribulose. It's used to generate ribose to make nucleic acids. Okay, it's very important for that and other metabolic products that we need. So, most, or pretty much every organism has got the pentose phosphate pathway. But in some bacteria, it's also used to generate energy, okay? Whereas for most of us, it's just used for making like ribose and nucleic acids. So if you kind of look at the yield of these 3 different glycolytic pathways, you get 1 or 2 molecules of ATP and 2 or 3 molecules of NADH. So you might think, well, that's great. We need both of these things. But there's a big problem for bacteria. And the problem is that in the biochemical economy of the cell, you need way more ATP than NADH. So in fact, glycolysis doesn't produce enough ATP compared to the NADH. And you might think, oh, well, that's no problem. We'll just run more glycolysis and get more ATP, right? But that won't work, because if you have more and more glycolysis, at the end, all the NAD plus is going to be used up. And the NAD plus is the essential cofactor for glyceraldehyde phosphate dehydrogenase, the enzyme that is catalyzing the essential step to get some energy out of this pathway. So you can't just run the glycolytic pathway faster and faster, because you'll use up all the NAD plus, and then the whole thing will stop. So in order to get it to run faster, you've got to be able to recycle these reduced forms of NADH into the oxidized NAD+. So you have to get rid of the electrons that are on NADH to regenerate the NAD+. So where can you put them? Okay, you've got to get rid of them somewhere. So what have you got left out at the end of your glycolytic pathway? You've got pyruvate, right? You started with glucose, you've got a load of pyruvate. You're not going to do anything with it anyway. Hey, let's just stick the electrons on there. Come on. You can have it. Pyruvate, have some electrons. Yeah, you like it. Come on. So, okay, that's the basic idea of fermentation, right? Has to get rid of the excess electrons on the NADH. And if that happens, okay, so this is a kind of schematic here. This is the glycolysis. You start with glucose, getting pyruvate, gives you NADH. You get a couple of molecules of ATP. But what you want is for any fermentation pathway, you want to get rid of the excess electrons here, regenerate the NAD+, and then it can be used up here again to fuel glycolysis. So if you just run this system, the NAD plus NADH just goes round and round and round. It's getting recycled. You crank it up, you can run it as fast as you like, and you'll get a load of ATP. Bingo! The problem is solved. So that's what the point of fermentation pathways is. Okay, get rid of the NADH so we can carry on generating ATP. So it allows continuous production of ATP by oxidizing NADH to NAD+. Now, well, in biology, nothing is simple, right? And certainly fermentation pathways in bacteria are not always simple. And there are a lot of bacteria that perform fermentation, and they can do it in a lot of different ways. But basically, the electron acceptor is always pyruvate or something derived from pyruvate. And the pyruvate itself is derived from the glucose that you used at the start of the pathway. So there is no external electron acceptor. Okay? Right, so let's have a look at some of these fermentation pathways. They all start from pyruvate, okay? So we'll start looking at the simple ones first, and then look at the more complicated ones afterwards. So one of the simplest is lactic acid fermentation. So you start with pyruvic acid, and lactate dehydrogenase will reduce this ketone group to a hydroxyl group, okay, to an alcohol group here. And that will give you lactic acid. So just look at the structure of lactic acid a little bit, okay? It's kind of an illustration of how fermentation works. Remember, the initial substrate is glucose, carbohydrate. So it's like if you broke this down into three glucose broken down into 3 carbons is what? Would be like glycerol, would be a 3-carbon sugar. I'm going to draw this out because otherwise I don't know if you'll get it. Okay, this is glycerol, 3-carbon sugar. Think about what's happened to this molecule to turn it into lactic acid. This part has become oxidized, become a carboxylate group, become oxidized. This part has been reduced because it's now become a methyl group. So that's why in the fermentation pathway, the carbohydrate that you put in, it's the electron donor and the electron acceptor at the same time. One part of the molecule gets oxidized and the other part gets reduced. And that's what gives you lactic acid. Well, that's my way of looking at it anyway. So lactic acid fermentation, very, very simple. Just requires one enzyme to convert pyruvate to lactate. And a lot of bacteria use this fermentation pathway. So lactobacillus, streptococcus, and plenty of others. So lactic acid fermentation can be homo or heterolactic. So homolactic fermentation, lactic acid is the only major fermentation product. Heterolactic fermentation, you get lactic acid plus something else. Now that's very useful for making things that we like to eat, right? So the first of the three examples here is making yogurt and fresh cream. I guess it would be called sour cream in English. This is my automatic translation of crème fraîche, sour cream. So if you get milk, doesn't keep very long, especially if you don't have a fridge. And the sugar in there is lactose. And if you inoculate it with starter culture containing two different lactic acid bacteria, Streptococcus thermophilus and Lactobacillus denbrookii subspecies bulgaricus. Often you call it Lactobacillus bulgaricus. Incubate them in your yogurt maker between 43 to 46 degrees, 3 to 6 hours, you get yogurt. So what's happened? Well, initially the Streptococcus thermophilus will grow, reduce the pH a little bit, and use up all the oxygen that's present. And then the Lactobacillus bulgaricus will start to grow, reduce the pH even more, and that's what will cause the proteins in the milk to coagulate and form this kind of gel texture that you have in the yogurt. Now, in order to get good yogurt, you need both of these bacterial strains. Okay, if you have just one out of two of them, it won't work. Okay, because if you've got only Streptococcus thermophilus, the fermentation will start, but the pH doesn't go down low enough to really precipitate out the proteins. If you've only got Lactobacillus bulgaricus, it doesn't grow very well at a neutral pH. So the fermentation doesn't really start. You need both of them at different stages in the process to get a nice yogurt. Okay, so if you make your own yogurt, you can always use whatever left of the one from the last time and use it to make the next round, because you'll have your mixture of lactic acid bacteria in there. Okay, another thing you can make from milk is if you have take off the cream, then inoculate it with Lactococcus lactis subspecies cremoris, and then a mixture maybe of Lactococcus lactis and leuconostoc. So these might be commensal bacteria that grow around the cow's udders, okay, where you get the milk from. So when milk is non-pasteurized, it will contain some of these bacteria. Now, fermentation here is at lower temperature, takes longer. Once again, you get kind of like coagulation. This will give you sour cream, crème fraîche. The pH is, once again, quite acidic. Now, some of the taste comes from the minor fermentation products that are produced by the leuconostoc. So if you don't have leuconostoc in here, you'll get something that looks the same, but it won't taste right. Okay. So that's how you make these things. Of course, another big dairy product is cheese, right? So once again, you start with milk, and you have the fermentation is initiated by what's called the starter lactic acid bacteria, or SLAB. Lactococcus lactis subspecies lactis and Lactococcus lactis cremoris. It's the same as these two, right? Same as these two. And once again, these are bacteria that are commensal bacteria that grow on the cow's udder. Everybody knows what I mean when I say that word, a cow's udder. Can anybody tell me what that is in French? Le pied de la vache, where you get the milk from, right? Okay, so you've got some commensal bacteria that grow there, and they contaminate the milk. non-pasteurized milk will contain these bacteria. So if you collect it very, very quickly before any other contaminant bacteria can come, and you just start the fermentation, then these were the ones that will mostly be present. So that's how, it's possible to make cheese with non-pasteurized milk, but you have to be careful. So if you use pasteurized milk, there's no bacteria in there. You've got to add them. Okay, you've got to add the starter lactic acid bacteria. So you add them, incubate 1 or 2 hours at 30 degrees C. It's fast. pH will go down rapidly, and then you add rennet, which is initially comes from the calf's stomach, and it will cleave the proteins and cause them to separate, some of them to precipitate out into the curds, le caillé, and the whey, which is the lactoserum. Okay, so when you make cheese, you get the curds, and you pick them out, and you press them, squeeze out all the liquid, and you mold it, wash it in salt water, so you increase the salt concentration. And then you form it into some kind of like a lump or a, kind of round shape. You put it in the cellar to, for it to mature for, could be months or a year. And this is kind of cold. Now, during this maturation process, there are still some bacteria that can live inside the cheese and carry on fermenting some of the products. And that's why the taste develops over time, because these non-starter lactic acid bacteria are producing minor fermentation products that gives the cheese, its flavor. Because if you just have this, you've got, like a fromage frais sort of thing, not much taste. Okay, so depending on this process and which non-starter lactic acid bacteria are present, you get different types of taste in your cheese. So, big example is Propionibacterium in Emmental. So these Propionibacteria, they can actually ferment lactic acid, which is here. They take 3 molecules of lactic acid and they will metabolize them into 2 molecules of propionic acid. So this is slightly reduced, right? 1 molecule of acetic acid and 1 molecule of carbon dioxide. So it's the same kind of thing. The lactic acid here, different parts of it are reduced and oxidized. So this is kind of reduced, and the CO2 is the oxidized form. So they can get some energy out of this, okay? And the CO2, this is the gas that gives you the bubbles in the Emmental cheese. So that's the non-starter lactic acid bacteria. One more thing to do with lactic acid fermentation. It's used for conserving vegetables, like in sauerkraut, Choucroute, and a lot of other, conserves that you can make at home. So what you do is you get your vegetable, slice it up, add salt. So the salt concentration's got to come up to about 20 to 30 grams per liter. And then you put it in the pot and leave it for several weeks, either at room temperature or slightly cooler. Okay, down, put it in the cellar. So you don't add any bacteria. So these are bacteria that are living on the surface of the plant. Now the salt here is important because you increase the salt concentration a bit to try and inhibit the growth of gram negatives. Because gram negatives, they don't like growing at low activity of water, whereas gram positives have a better survival. Also, put it in the pot, all the oxygen is going to be used up very quickly. So very quickly, there's no more oxygen, and the only way bacteria can live is by fermentation. So they'll ferment this, whatever kind of carbohydrates they find in the plant, and that will turn the whole of the medium very, very acidic. So this is from a paper that I found, published last year, where they looked at the bacterial populations present in homemade preparations of fermented vegetables. So you could see that, well, whatever the vegetable was used, The pH is very, very acidic, goes down to about 3.5. And you can have, quite a lot of lactic acid bacteria per ml. So about a million per ml most of the time. And of course, these bacteria, they're still alive, right? So when you eat your homemade choucroute or your whatever conserved carrots, then you're getting a lot of lactic acid bacteria. So here, there's no specific bacteria that are added at the start of the process. So what these researchers were interested in was finding out what are the bacteria involved. And so they had, like, most of the time, these 3 different genera, which are called Lactobacillus, Lactoplantibacillus, and Lactobacillus. So this one is a recent name. Normally, if you look this up in sauerkraut, it will say that it's caused by Lactobacillus plantarum. So this is the previous name of what is now called Lactoplantibacillus, not Lactobacillus plantarum. So you have this mixture of these lactic acid bacteria and other things like Leuconostoc again, Weissella, this kind of thing. Okay, so once again, it's a food conservation technology, because you can't keep something like this on the shelf for very long. So if you've got a big harvest of cabbage or something, and you don't have a freezer or a fridge because it's the 19th century, nobody's invented it yet, you have to conserve your food with something like this. And it basically will work. You just need a bit of salt and a bunch of pots, and you can keep whatever you've got for like several months. And it'll keep you alive through the winter. It's a very, very important thing. Okay, I'll try and do one more fermentation. We've got 8 minutes. We can do alcoholic fermentation, I think. Okay. So once again, simple fermentation pathway. It starts off with pyruvate. The first step is pyruvate decarboxylase, which will remove one molecule of carbon dioxide. That gives you acetaldehyde, which will then be reduced to ethanol. So the reduction step, this is what fermentation is for, is to remove the electrons from the NADH and put them somewhere else. So this is alcohol dehydrogenase, which does this. Okay, so this is, of course, everybody knows yeast does this. That's what we use to make wine, beer, all kind of fermented alcoholic drinks. But also some bacteria can perform alcoholic fermentation, including this gram-negative called Zymomonas mobilis. So I'm not really going to talk about wine, brewing, this kind of thing. So that's yeast stuff. It's not bacteria, but it's important. But outside of the food industry, there's kind of an interest in alcoholic fermentation to produce what's called, bioethanol, or more like agri-ethanol, right? To try and produce ethanol for car fuel from agricultural waste. So the idea is you get like your agricultural waste, in a field of maize, for example. You get the grains, you can eat them, or often they're given to pigs for food. But you've got the rest of the plant, which is just like lying around in the field. So if you harvest that, then you can grind it up. So in the gray here, this is a physical process. Blue is a kind of biochemical process, and orange is my microbiological process here. So gray, the physical process, you grind it up into some kind of feedstock. Now at this point, you have cellulose fibers. And not all bacteria can degrade cellulose. So you have to put some enzymes in there that can break down the cellulose into the sugars. So break down the cellulose fibers, and then you can put this into your fermentation system with yeast or Zymomonas mobilis. And then you'll get the sugar that was from the cellulose, from the plant tissue, will be converted to alcohol. Then you filter this, bacterial culture, get rid of all the solids, get the liquid, distill it, and then you get agri-ethanol. So there are, there's industrial applications for this alcoholic fermentation. Now, I don't want to talk about this. I think I might go forward and talk about something more fun, actually. So, yeah, I'll just tell you this one and then we'll stop, okay? So, alcoholic fermentation can also occur in our commensal bacteria sometimes, or commensal yeasts that live in our gut. And in fact, it's happening all the time at a low level. So that's why, you can never have a lower limit for your, if you get tested, you're driving your car and you get taken to give a urine sample or blood sample to measure the alcohol level in your blood. There's always a little bit in there, and it's being produced by bacteria in your gut. And sometimes this can cause a real problem. So this was an example from just, it was in the newspaper just this spring. It was published in West France in April. It's a story about this person in Belgium. He was stopped and tested by the police because he was driving all over the place like this. And he had a 0.9 milligram per liter of alcohol in the breathalyzer test. That's way over the limit. Months later, stopped again, 0.7 milligram per liter. I said, okay, that's it, come on. And that's it. Your license, that's it, you're finished. And I think he got maybe even perhaps a jail sentence or something. But so he contested it. He said, no, I haven't been drinking alcohol at all. It's not true. I haven't been drinking anything. And so he got a medical expertise, and he was right. He was convinced, yeah, I can't have alcohol because I haven't had anything to drink today. And he was telling the truth. And it's because he was suffering from this auto-brewery syndrome. So there are sporadic cases, and it's because you've got Candida or Saccharomyces living, in your gut. And when you eat something which is sugary or starchy, they can ferment it into alcohol. So it's mostly yeast, commensal yeast living in your gut, but it can be caused by mixed acid fermentation by some bacteria of the Klebsiella genus. So what are the risk factors for this? Type 2 diabetes, if you've got a starchy or a sugar-rich diet. If you have a big meal full of a load of sugar and a lot of pasta or something, then if you've got these yeasts or Klebsiella in your gut, then they can start fermenting and producing alcohol. The other risk factor is genetic deficiency in alcohol dehydrogenase. Okay, so this is the enzyme in the liver which will detoxify ethanol. It's just the same one as used in alcoholic fermentation, but running in reverse. It will convert ethanol into acetaldehyde using NAD plus as a cofactor. That's right. So most people in Europe, we have alcohol dehydrogenase, right? But where do people have not so much? Can't take a drink, get drunk very quickly. Bit of a racial stereotype, but it's because of this. Oh, come on, you must know. No? Okay. There are a lot of people in Japan, maybe other East Asian countries, who lack alcohol dehydrogenase. Okay. And in that case, this kind of auto-brewery syndrome is more frequent. It's actually published years ago in the medical literature in Japan. People driving around in their car, they're stopped by the police, they've got an alcohol, and in their blood, and it's come because they've had a meal with a lot of rice, and the gut bacteria are fermenting it, and gut yeasts are fermenting it into ethanol, and because they don't have alcohol dehydrogenase, for us, right, the alcohol is going to be absorbed, it's going to go into what? Blood vessel? Anything you absorbing the gut goes into what blood vessel? Hepatic portal vein, a vein porte. And then it goes to the liver, right? So that's the first stop. Everything that comes from your gut goes to the liver. So if you've got alcohol dehydrogenase, then this fermentation, if it's at a low level, is not going to cause a problem. But if you're deficient in this enzyme, if you have this alcoholic fermentation going on in your gut, you're going to get the full dose in your bloodstream. So it's more of a problem with people who have alcohol dehydrogenase deficiency. And so the only thing you can do, try and avoid this kind of diet. It's the only thing that can really happen here. Okay, so thanks everybody for listening. I'll let you go. And microbe of the week, Sunday, 7 p.m. Is that okay? I will try and send it to the right people this time.